Hi. Today we are excited to present our project on creating a biosensor for diagnosing breast cancer. Credit goes to Vikas, Samhita, and Nandana for their contributions. We hope you find our work informative and inspiring. Before looking into anything else, we should first understand the background of breast cancer and why we want to work on it. Breast cancer has become so common that every 1 out of 8 women is diagnosed with breast cancer. According to recent global cancer estimates from the International Agency for Research on Cancer, IARC, female breast cancer has become the most commonly diagnosed cancer type in the world. In 2020, approximately 2.3 million women were diagnosed with breast cancer, surpassing the number of new cases of lung cancer for the first time. Breast cancer caused about 685,000 deaths in 2020, making it the most common cause of cancer death in women and the 5th most common cause of cancer death overall. Deaths occur annually, among which cancer deaths are one of the highest after cardiovascular diseases. The data for India indicates that breast cancer cases have increased from 1900 to 2019. Among all types of cancer, breast cancer cases are the highest, as depicted in the image. Cancer develops when cells' DNA is damaged. The reasons for why and how DNA is damaged are still unknown, but there can be multiple factors contributing to it. Mutations in BRCA1 and BRCA2 genes impair DNA repair, leading to a higher risk of breast and ovarian cancers. While not everyone with these mutations develops cancer, their presence substantially increases the risk compared to those without them. These genes are crucial for maintaining genetic stability and preventing the accumulation of mutations that can contribute to cancer. Several factors are associated with breast cancer, such as 1. Family history. If anyone in the family has had breast cancer earlier, it can increase the risk. 2. Early menarche. Onset of menstruation before age 13 is a risk factor. 3. Late menopause. Menopause occurring after age 55 is another risk factor. It's important to note that while these factors are associated with an increased risk of breast cancer, not everyone who has these factors will develop the disease. Conversely, someone without these factors can still develop breast cancer. This highlights the complex nature of the disease and the need for comprehensive understanding and research. BC team members surveyed reported experiencing stress, depression, or anxiety related to breast cancer. Many noted negative effects on their family due to breast cancer. Breast cancer patients face physical side effects, emotional impact, body image, and sexual health concerns, as well as social and economic challenges. Despite decreasing mortality rates thanks to improved detection and treatment, the primary concern now extends to long-term quality of life, emotional well-being, and effectively managing the chronic aspects of survival. Here are the symptoms of breast cancer: new lump or mass in the breast, swelling of all or part of the breast, skin irritation or dimpling, breast or nipple pain, nipple retraction, turning inward, redness, scaliness, or thickening of the nipple or breast skin, nipple discharge other than breast milk, change in breast color, pitting of the breast skin. Let's look into the available diagnostics and prognosis tools for breast cancer. Existing diagnostic techniques for breast cancer are utilized when symptoms or abnormalities are present. 1. Mammography, cost 4,000 Indian rupees. This imaging method captures X-ray images of breast tissue when visible abnormalities are present, aiding in diagnosis and assessment. 2. MRI, cost 8,000 Indian rupees. Magnetic resonance imaging is employed to visualize breast abnormalities and assess their nature, particularly helpful in cases with suspected tumors or complex presentations. 3. CT scan, computed tomography, cost 5,000 Indian rupees. CT scans are used to detect and evaluate breast lesions or abnormalities once they are identified or suspected. 4. PET scan, positron emission tomography, cost 15,000 Indian rupees. PET scans are utilized to assess metabolic activity in breast tissue, providing additional information in cases of suspected cancerous growths. 5. Ultrasound, cost 2,500 Indian rupees. Ultrasound imaging is employed to examine breast abnormalities such as lumps or changes in tissue, aiding in diagnosis and guiding further evaluation or biopsy procedures. Molecular tests are also used in breast cancer diagnosis and treatment, although they are relatively expensive. MammaPrint, $4,200, provides genomic analysis to predict breast cancer recurrence risk. ER/PR expression costs 6,000 Indian rupees, measures estrogen receptor ER and progesterone receptor PR levels in breast cancer cells. HER2 overexpression costs 6,000 Indian rupees, assesses the amplification of the HER2 gene, which can influence treatment decisions. Gene panel testing costs 2,000 Indian rupees, analyzes multiple genes related to breast cancer to assess risk and guide personalized treatment. Oncotype DX, $3,500, evaluates the activity of certain genes to predict the likelihood of breast cancer recurrence. Prosigna breast cancer assay, $3,000, assesses the risk of recurrence in hormone receptor positive breast cancer patients. Germline testing cost 16,000 Indian rupees, identifies inherited mutations in genes like BRCA1 and BRCA2, which can increase breast cancer risk. Additionally, various biosensors are being developed, although they are currently limited to laboratory settings. Our next goal is to identify a pressing issue in the market that requires an urgent solution. We will delve into the various aspects of the problem to address the issue of breast cancer in society. First, the number of breast cancer cases is increasing significantly, impacting a large segment of the population, especially women. Women in rural areas rarely undergo breast cancer screening. Secondly, the critical issue is at what stage we can detect the disease. Sometimes, detection occurs too late, at the final stage when metastasis has already begun. Thirdly, there is a problem with the biomarkers we use to diagnose this disease. Fourthly, there is a significant gap between the existing solutions for breast cancer and what is actually required for the accessibility of the population. During our research, we identified several problems and gaps in the early diagnosis of breast cancer cases in India. The affordability of gold standard diagnostic methods is a significant barrier, limiting access to timely and accurate detection. Additionally, there is a lack of market availability for point-of-care tests, POCs, that are both affordable and effective. The presence of multiple breast cancer subtypes and the variety of biomarker options further complicate diagnosis and treatment, with very few specific markers available for multiplexing. Recognizing these challenges, we focused on developing a biosensor intervention that could be used for both prognosis and diagnosis, aiming to provide a cost-effective, accessible, and reliable solution for early breast cancer detection. Let's delve into the gaps in existing diagnostic solutions for breast cancer. In terms of imaging techniques like mammography, CT scans, and PET scans, although they're considered gold standards, they come with drawbacks. They are very expensive. They use ionizing radiation, which can be a concern. They are time-consuming, taking about 3 to 4 hours for a session. They lack early detection capabilities and have lower resolution compared to newer technologies. Moving on to molecular testing methods such as MammaPrint, Oncotype DX, and Prosigna breast cancer assay, while they provide valuable information, they also have limitations. They require invasive procedures like biopsies. They are quite costly, ranging from $3,000 to $4,200 per test. Insurance coverage for these tests is often limited. The turnaround time for results can be as long as 2 days. Now, let's discuss biosensors developed in research labs. Despite their potential, they face challenges that prevent them from reaching the market. The process of immobilizing enzymes for these biosensors is costly. Gold nanoparticle labeling, although effective, adds to the overall cost and can be affected by label instability. Real sample investigations, crucial for accuracy, are often lacking. There's a need for multiplex detection to cover a wider range of breast cancer biomarkers effectively. Lastly, when considering point-of-care POC devices for breast cancer, we encounter significant gaps. Affordable POC devices are not yet available. Even the developed POC devices in research labs have limitations, including low signal-to-noise ratio, the need for expert sample preparation, and the absence of multiplexing capabilities. These gaps collectively emphasize the urgent need for more accessible, efficient, and cost-effective diagnostic solutions for breast cancer. Let's look into the problem with biomarkers. These biomarkers from samples such as blood, serum, and urine are non-invasive. However, the sample used for molecular testing, which is taken from a biopsy sample of breast tissue, is extremely invasive. Levels of estrogen and progesterone are detected in samples taken from breast tissue using invasive methods. Upregulation of ER indicates a higher risk of developing breast cancer. Mutations in BRCA1 and BRCA2 can be detected using a blood sample. Now let's delve into the solutions we can develop for the aforementioned problems. It's important to note that designing solutions that address all the discussed problems is extremely challenging. Therefore, we will focus on those problems that are most critical and truly require solutions. To create a solution space, the initial step involves determining if an individual has breast cancer. If diagnosed, the subsequent step is to ascertain the severity and type of cancer. There are 4 types of breast cancer: luminal A, 50% of cases; luminal B, HER2, and TNBC, 15% of cases. Initially, it might be unclear which type to test for. Therefore, we have included biomarkers for all 4 types, multiplex assay development, to diagnose breast cancer and determine the specific type. Hence, multiplexing is a necessary requirement. We have decided to focus on developing a biosensor for diagnosing breast cancer rather than predicting its progression. The reason for this choice is that predicting the progression of breast cancer is more complex compared to diagnosis. This complexity arises from the diversity of biomarkers introduced as the disease progresses. For gender inclusivity, it's important to note that while 60% of women aged 40 and above undergo mammograms, 1% of men are also affected by breast cancer. Our solution is to create a biosensor capable of effectively detecting breast cancer in both men and women. However, a potential problem we may encounter is the variability in the level or expression of biomarkers associated with breast cancer in both genders. Affordability is a crucial aspect of breast cancer screening, considering that mammography, the gold standard, costs $4,000 per test, while the average monthly salary of an Indian citizen is $8,000. Our solution is to develop and validate a point-of-care POC screening test that is affordable for all socioeconomic classes in India. The type of biosensor intervention we aim for is a point-of-care PO screening test targeting early diagnosis of breast cancer. Immunohistochemistry and molecular assays, which are currently available but expensive in diagnostic labs in India, inspired our approach. Early diagnosis is crucial as 50% of breast cancer cases, specifically luminal A subtype, could be prevented from progressing or resulting in death with accessible early diagnosis. Now let's look into how we made the choice for a biomarker to detect and diagnose breast cancer in patients. 1. Luminal A. This is the most common subtype, making up 50% of all breast cancer cases. These tumors are positive for estrogen receptor, ER, and progesterone receptor, PR, but do not express HER2. They have specific microRNAs, miRNAs, and typically have a good prognosis. Treatment usually involves hormonal therapy. 2. Luminal B. Accounting for 15% of cases, luminal B tumors are also ER positive, but may or may not be PR positive. They do not express HER2 and have different miRNAs compared to luminal A. These tumors have a higher proliferation rate, poorer prognosis than luminal A, and may require additional treatments along with hormonal therapy. 3. HER2 positive. About 20% of cases fall into this subtype, characterized by overexpression of the HER2 protein. These tumors may or may not be ER or PR positive and have unique miRNA profiles. They typically have a high proliferation rate, and targeted therapies against HER2 have improved prognosis significantly. 4. TNBC, triple-negative breast cancer. TNBC accounts for 15% of cases and does not express ER, PR, or HER2. It has different miRNA profiles and a high proliferation rate, making it more difficult to treat. TNBC does not respond to hormonal therapy or HER2 targeted drugs, leading to a poorer prognosis. Each subtype has its characteristics, treatment options, and prognosis, emphasizing the importance of identifying the specific subtype for effective management of breast cancer. Luminal A breast cancer is characterized by elevated levels of estrogen and progesterone receptors, indicating its hormone-sensitive nature. In contrast, luminal B subtype exhibits elevated levels of estrogen receptor along with the presence of BRCA2 mutations, which can influence treatment strategies. The HER2 positive subtype is identified by elevated levels of HER2 and Ki67 proteins, highlighting its aggressive nature and potential responsiveness to targeted therapies against HER2. On the other hand, triple-negative breast cancer, TNBC, is distinguished by elevated levels of Ki67 and the presence of BRCA1 mutations, contributing to its high proliferation rate and challenging treatment options due to the lack of targeted therapies for hormone receptors and HER2. These distinct molecular profiles underscore the importance of precise subtype classification for tailored treatment approaches in breast cancer management. When developing a multiplex assay, the goal is to detect all nucleic acids or proteins using a single assay. However, in our case, we encounter both nucleic acids and proteins as potential biomarkers. Notably, the estrogen receptor ER and progesterone receptor PR cannot be detected in blood samples. They are tissue-specific and can only be obtained from breast tissue. Therefore, ER and PR are not utilized as biomarkers in our assay. Similarly, while P53 is highly specific to cancer, the BRCA1 and BRCA2 genes present numerous mutations, making it challenging to pinpoint which mutation causes breast cancer. This uncertainty regarding the specific mutations in BRCA1 and BRCA2 makes them unsuitable as biomarkers for our assay. Additionally, even if a mutation is present in BRCA1 or BRCA2, it does not guarantee the presence of breast cancer. Consequently, BRCA1 and BRCA2 mutations are not included in our biomarker panel. Instead, we focus on the upregulation and downregulation of miRNA as potential biomarkers for diagnosing breast cancer at different stages. Despite various papers suggesting different markers for early cancer detection, we select miRNA biomarkers based on the reliability of the journal and the publication date of the papers. There are many biomarkers related to breast cancer. Pinpointing the miRNA to be used for detecting breast cancer was not an easy task at all because many miRNAs were expressed tissue-specifically in the breast. Therefore, we decided to choose circulatory miRNA as our biomarker. Circulatory miRNA is advantageous because it can be found in blood samples, allowing us to use blood as our sample for detection. Many miRNAs are upregulated or downregulated not only in breast cancer, but also in other types of cancer, such as cervical and prostate cancer. For example, miRNA 21 is common in all three, and using it would decrease the specificity of the assay. Therefore, we have not chosen miRNA 21 as it is not very specific to breast cancer only after reading many papers. We have selected specific biomarkers for different subtypes of breast cancer to enhance the accuracy and sensitivity of our assay. For luminal types A or B, we have identified miRNA 1290 as a key biomarker. In HER2-positive breast cancer cases, we have observed a downregulation of miRNA 195 and an upregulation of miRNA 146A-5P, which are indicative markers for this subtype. Additionally, for triple-negative breast cancer, TNBC, we have noted an upregulation of miRNA 20A and miRNA 92A. By using a combination of these miRNAs, we aim to increase the sensitivity of our assay significantly. This approach allows us to detect breast cancer more accurately. After extensive research and effort, our final achievable goal is to develop an affordable point-of-care biosensor capable of detecting multiple biomarkers. This biosensor will be designed to differentiate between luminal AB, HER2, and triple-negative breast cancer subtypes. Our objective is to ensure accessibility by making this technology widely available and affordable for healthcare providers and patients alike. By enhancing early detection and facilitating personalized treatment strategies, this biosensor aims to improve outcomes and save lives among breast cancer patients. Once we are clear about our problem statement, let's look into how we can work on it. To develop our device, we first need to extract our biomarker from the blood samples of the patients. Before diving into the exact miRNA extraction technique, we need to pre-process the blood samples. For this, one has to take approximately 150 millimeters of the patient's blood. Then, we need to separate the plasma from it. For separating the plasma in low-resource settings, researchers have developed a paper-based technique. As shown in the image, to separate the plasma, one has to put the sample on a paper-based device with a specific design. Keep it horizontally for 3 minutes, and after that, keep it against gravity. This technique has been used before, so we assume it will work for us as well. This is the pre-processing part of the sample. After that, place the blood in a polytetrafluoroethylene PTFE tube. This tube is resistant to the chemicals we are going to use for the extraction of miRNAs. When the blood is introduced into the tube after breaking the valve between the tube and the paper-based sheet, all the extracellular miRNAs will bind to the magnetic beads. These beads have a silica coating that is positively charged, allowing them to bind with the negatively charged nucleic acid backbone. In the first chamber of the tube, proteases are added to degrade all the proteins present in the plasma, ensuring they do not affect the subsequent processes. We then move the magnetic beads, which are attached to all nucleic acids, to the next chamber, the wash chamber. Here, an RNase inhibitor is added to prevent RNase from degrading our miRNAs, and DNase is added to destroy the circulating DNA. After allowing time for these enzymes to act on their substrates, the beads are moved to the final chamber, the elution chamber, where the miRNAs are eluted from the beads. This movement of the beads can be repeated 2 to 3 times to ensure that all miRNAs are extracted. The complete process will take approximately 30 minutes in total. After extracting the biomarkers, our next step is to amplify these miRNAs. We choose to amplify miRNAs because existing literature shows that in breast cancer patients, even during early detection, specific miRNAs can be either upregulated or downregulated, sometimes at levels as low as 0.5 femtomolar. The detection technique we are using has a limit of detection of 0.25 femtomolar, making amplification crucial to enhance detection sensitivity. Once all miRNAs are in the elution chamber within the same tube, we can facilitate amplification by adding an additional chamber specifically for this purpose, with a valve between the elution and amplification chambers. By pressing a button, we can transfer the eluted material into the amplification chamber. Here, we will employ exponential amplification reaction, XPAR, an ultra-sensitive technique designed for miRNA amplification. XPAR can amplify concentrations ranging from zeta-molar to micromolar, achieving 10 raised power 6 to 10 raised power 9 copies per milliliter. XPAR operates under specific conditions, including a temperature range of 61 to 65 degrees Celsius and a reaction time of 30 minutes. It amplifies the miRNAs such that our detection technique can detect these miRNAs in picomolar concentrations. XPAR's detection window spans from picomolar to micromolar, with maximum sensitivity down to 0.1 zmol 100 copies per 10 micrometer reaction. The reaction mechanism involves cycles of binding, extension, nicking, and strand displacement, leading to exponential amplification of the target oligonucleotide. This setup ensures that our detection strategy can effectively identify even trace amounts of miRNAs, enhancing the sensitivity and reliability of our biosensor for breast cancer detection. Amplification is done. Our next step is to detect the complementary products of the miRNAs. For this, we are using a sensitive and highly specific technique known as bioluminescence resonance energy transfer, BRET. BRET allows for precise detection of the amplified miRNAs, ensuring accuracy and reliability in identifying the specific miRNAs related to breast cancer. This technique's sensitivity makes it ideal for detecting even low concentrations of miRNAs, enhancing the overall effectiveness of our diagnostic approach. This detection technique is paper-based, previously utilized by researchers. The results appear directly on the paper, making it very specific due to the use of zinc finger proteins, which bind to specific DNA sequences. The principle behind this technique involves one donor molecule and one acceptor molecule. These molecules are attached to zinc finger proteins, which bind to specific sites on the DNA molecule. When these two molecules bind to their respective sites, the donor and acceptor molecules come together, resulting in an exchange of energy and a color change from blue to green. The intensity of the color indicates the concentration of the miRNAs, and a smartphone can be used to measure this concentration gradient. The limit of detection for this technique is 5 into 10 raised power minus 3 picomolar, and it works within a range of 5 into 10 raised power minus 3 to 50 picomolar. Now we need to design zinc finger proteins specific to the miRNAs we are targeting. This is how this detection technique can be utilized to detect multiple biomarkers. As our miRNAs of interest are amplified in the amplification chamber, we can press the valve button again, allowing the miRNAs to travel through microfluidic channels to the specific paper where the detection reagents are already present. We also include a positive control for each miRNA, which is synthetically designed and kept on the paper. As soon as moisture reaches the positive control, it will ensure the assay's reliability. The color change on the paper indicates the concentration of specific miRNAs, which can be scanned by a smartphone. This is how we can quantify the biomarker availability in the patient's sample. This is the overall proposed solution of our project, ideated by our team. First, we focus on blood sample collection and pre-processing, where approximately 150 milliliters of the patient's blood is collected. Plasma is separated using a paper-based technique suitable for low-resource settings, and proteases are employed to degrade proteins, ensuring the plasma is free from contaminants. For miRNA extraction, the blood sample is transferred to a PTFE tube resistant to the chemicals used, and magnetic beads with a positively charged silica coating are introduced to bind extracellular miRNAs. Proteases in the first chamber degrade proteins to prevent interference with further processes, and the magnetic beads are moved through wash and elution chambers to isolate and purify miRNAs. The purified miRNAs are then transferred to the amplification chamber using microfluidic channels, where exponential amplification reaction, XPAR, is utilized to amplify the miRNAs with high sensitivity, achieving final concentrations in the picomolar to micromolar range within 30 minutes. For miRNA detection, a paper-based bioluminescence resonance energy transfer (BRET) technique is used. The sample is applied to the paper, where zinc finger proteins bound to donor and acceptor molecules cause a color change from blue to green, indicating the presence and concentration of miRNAs. A positive control on the paper ensures assay reliability, and the color intensity is measured using a smartphone to quantify the miRNA concentration. There are a few limitations to our solution that need to be addressed. First, the screening and selection of specific zinc finger proteins, ZFP, is both expensive and time-consuming. Identifying the right ZFPs that can accurately bind to the target miRNAs requires extensive research and validation, which can be resource-intensive. Second, the synthesis of the selected ZFPs is costly. Once the appropriate ZFPs are identified, producing them in sufficient quantities for our detection technique involves significant expense. Additionally, integrating a machine learning ML model into our solution requires more data points, which will take time to gather. The ML model is crucial for improving the accuracy and reliability of our detection strategy, but it needs a large data set to learn from and make precise predictions. Moreover, the combined XBAR and BRET strategy has to be tested in the lab. Although this approach shows promise, it is essential to validate its effectiveness through rigorous laboratory experiments to ensure it works as intended. Furthermore, optimization and testing using clinical samples are necessary to map fluorescence to the concentration of miRNAs accurately. This step involves using actual patient samples, fine-tune our detection method, and confirm its clinical applicability. Lastly, we need to optimize the plasma separation paper-based method. Currently, 3 milliliters of blood yields only 150 micrometers of plasma, and improving this process is crucial for enhancing the overall efficiency of our solution. Despite these challenges, our proposed solution offers a comprehensive approach to the detection of miRNAs associated with breast cancer. Addressing these limitations through further research and development will help realize its full potential. In this project, we began by identifying breast cancer as the target disease and reviewed existing diagnostic solutions, including mammoprint, mammography, MRI, and biosensors, noting their high costs and affordability issues. We then focused on miRNAs as the biomarker due to their ability to provide a cost-effective solution that can detect multiple biomarkers and differentiate between breast cancer subtypes. Our strategy for isolating and processing miRNAs involved extracting them from plasma using magnetic beads, followed by amplification using the exponential amplification reaction, XPAR technique. Finally, we employed bioluminescence resonance energy transfer (BRET) as the detection strategy to complete our diagnostic approach. We're looking forward to advancing this project and developing the full device in the future. Thank you for your time, and please write in a comment if you have any doubts, feedback, or anything to share with us.